right. So, I first want to know who you are, what is your name, which faculty you study in, because I want to change the course according to what you know. Okay. So please let me know, what grade you are, what is your faculty, what is your name, and why you take this course, what you want to learn. Okay, let's start like this. I will close the door. So let's start.
My name is Hidekada. I'm first grade, and I belong agriculture.
Agriculture.
And I want to study about body and how to work how to relate each cells.
I see.
In our body.
Right. Okay. So, did you have biology in high school?
No, and I study physics and chemistry.
No biology.
Yes.
Okay. Thank you very much. Next.
My name is Yuki Toriba. I'm first grade, and I'm from Human Health Sciences.
Right.
I want to study biology in English for future laboratory works.
Okay. So you already have some biology knowledge.
Probably yes.
Okay.
Use biology in entrance exam.
Okay. So maybe some of the things that I will teach you, maybe you already know. This is possible. But in English, it may be different. Okay.
Yeah.
Next.
My name is Seiko Matsunari. Seiko Matsunari. I'm from Health Science.
Right.
I'm first grade. I want to study cell biology in English.
All right.
I study biology in high school.
I see. But of course, you studied in Japanese, right? And then you want. So you want to learn the subject, but you want to learn the language. Okay. What about behind?
My name is Masahiro Nambu. I belong to Faculty of Agriculture.
Okay.
So I didn't study biology in high school, but I need to study biology because I am a student of the Faculty of Agriculture.
I see.
So I choose this course.
All right. Okay. Next.
My name is Kazuyoshi Watanabe, and I'm in agriculture faculty, and I'm in second grade.
Second grade.
Yes. And why I choose this is I haven't studied biology in high school, but we use biology in my faculty, so I want to learn basic biology.
I see.
In this class.
Okay.
Sorry.
No problem. No problem. Please have a seat. What about yourself? Fourth grade. First grade.
biology.
I see.
So,
Okay, わかりました。Okay. What about our last person? So I want to know what year, what is your faculty, why you take this course, what you want to learn. So please let us know.
Okay, first of all, my name is Farhan.
Mm-hmm.
I'm a Queen student, exchange student.
Right.
Currently, I'm in my 10th academic year.
Okay.
I'm majoring in applied physics.
Applied physics.
Back in my home country, and I'm just basically in Kochi University. I'm doing random stuff.
Okay.
Yeah, and I'm taking this basic biology and metabolism to learn more about biology.
I see. Okay. All right. So I think more or less everybody is kind of similar. Even if you come from different faculties, you want to learn basic biology in English. All right. So I'll just present myself. So I am Jean-Michel Fustin. I am not English. I am not American. I am not from New Zealand. I am not from. I'm not from Australia. I am from Belgium. Right? I am from Belgium. But I studied about. Please, have a seat. Please let us know who you are, what year you are in, and why you come to this class. While you sit down, please. Please, let us know.
My name is Hajime Neo.
Mm-hmm.
I come here to study in this class.
Why? Yeah, many courses. Why you chose this one? What particular thing in the syllabus was interesting? Why were you interested by this?
Because I like biology.
Okay. And which faculty are you from?
I am from
Don't worry. Go slowly. That's no problem.
science faculty.
Science faculty. Nano science faculty. What is your main subject?
My main subject is I don't have my main subject
Okay.
because I don't have a main subject
Okay.
yet.
Have you studied biology before?
I studied biology one year ago.
1 year ago. Okay, alright, so please have a seat. So as I was saying, yes, so I am originally from Belgium, but I studied in Scotland, in the United Kingdom. And after my PhD in Scotland in 2008, I came to Japan as a postdoc. And then I became lecturer here. And now my research is circadian biology, Jikanseibutsu. And this is my main subject. But I also teach basic biology, chronobiology, and I also teach English in this faculty. So my hope is that because I am not from America or England, you will not be afraid of speaking English. Because I understand. I was a student like you, and I was not, of course, speaking Japanese. I was speaking French. But for science, I had to learn English. So I understand what you all feel. All of us, our main language is not English. So please don't be afraid, okay? And whenever you want to get more information, whenever you have a question, please raise your hand and ask a question. Do not be afraid. Do not leave the question at the end of the class. Because usually I believe that questions have to be asked immediately. When you have the question, you must ask within the subject. Because after, at the end of the class, I think it is too late. Okay? So please do not hesitate. I want to establish a discussion between us. Not just me always talking. That's not so interesting. Okay? Right. So I have made some slides already. Some of you may drop and not follow this lecture anymore, but anyway, I made some slides. I made some slides for you today. So you can at least have the beginning of the chapter. Staple them, but always time is an issue. Right. I hope I have enough. Right. I also put all these slides on Culasys, so you can download them if you want. On Culasys they are bigger. I have printed them bigger. So whenever you want, you can download. There is one here. And I do not have enough. So, well, you can download them on Culasys, okay? Right? So, but today is just introduction, okay? So don't be afraid. And I will go very slowly because it's the first lecture. First, I want you to tell me, what is life? Tell me a word. I just need one word from you. What is life? What characteristics? Think about it. If you think, okay, I am alive. My cat is alive. My fish is alive. But, my tree is alive. In my garden, my vegetables are alive. But, what is life? What are the characteristics of life? What makes us alive? What makes a bacteria alive? What makes a virus alive? Although that's questionable. Can you tell me some words that you think is important for life?
Self-replication.
Replication. Yes, self-replication. All living things, in the right circumstance, will replicate. We want to make more of ourselves. We want to have kids. Bacteria want to have sister bacteria, daughter bacteria. Yes, this is very important. But it is not enough. But it is not enough.
Growth.
Growth, yes. Growing is also important. Most organisms, they start in kind of a very early stage and then develop, especially more complex life forms, they will grow. This is also very important. What else? I can think of one more. Sometimes it's not very obvious, but it is also important. So reproduction, growth.
Take energy and use it.
Yes, exchange of energy and matter with the environment. This is true. An interrelationship with the environment. This is very true. You had also one. Go.
I have no idea.
No idea. I can think of one more. Exchange of matter and energy, reproduction or self-replication, growth, and what else? Think. Sometimes it's difficult, but if you actually look with a microscope, you will see that this really happens. Or if you look for long enough, you will actually see happening. It is a little bit sometimes similar to growth in a way. This is very different from like looking at a bench, for example. Movement, yes. Movement is also important. Most living things move. Right? Mostly bacteria moves. A tree moves. You don't see it, but a tree moves. Okay? A plant will move. And if the tree doesn't look like it's moving, you can look inside his cells, and then you will see a lot of movements inside the cells. So these are the main, really, characteristics of life. And it's really amazing that if you go down to the single cell, one cell has all these characteristics. A cell will grow. A cell will move. A cell will reproduce itself, cytogenesis. It has all the characteristics of life. And because of that, a cell is considered one unit of life. Okay, like 1 gram is a unit of weight, but one cell is 1 unit of life. Okay? Protein, this is not maybe much alive. Some proteins move, but they do not self-replicate. They do not necessarily grow. Okay. DNA might have the capacity to replicate, but not by itself. It needs many other things. So really, a cell is really considered as one unit of life. You could say, what about virus? What about virus? Virus is more difficult. What is a virus? What is a virus composed of? Well, there are many different families of viruses. What does usually a virus contain? Well, you don't see, but if you see a virus, what does it contain? What are the main constituents of viruses? What are the main constituents of viruses?
DNA and sugar.
Yeah, DNA, it really depends the virus. So I would maybe say nucleic acid.
Nucleic acid.
Because some viruses, they do not have DNA, they have RNA. Their genome is RNA, not DNA. And some viruses have sugar, but usually it's mainly protein and nucleic acid. But a virus is a kind of very strange because a virus does not reproduce by itself. A virus is just a bag containing RNA or DNA. And if you leave a virus on the bench, it will never do anything. It needs a cell to become alive. A virus will inject its genetic material into a cell and will hijack the cell mechanisms to make more copy by itself. So a virus is a very strange thing. Some people say viruses are not really alive because they need a cell to really have the characteristics of life. So a cell is really this living thing. Okay? So this introduction today is about cell. What is the very general structure of the cell? What is the origin of the word? Do you know why a cell is called a cell? Have you ever wondered why a cell is called a cell? This is the kind of things I will tell you. Cells, when we have cell in our mind, we can see maybe this textbook picture with we have a cell and a nucleus. But actually cells have very different appearance and function. All these looking very strange. And misshapen because I need to change my screen resolution. Okay. That's oh my God, what's happening? This never works like you want it to. Oh, well, that's okay. Look at it. Okay, these are all different cells. These are all different single cells. You have a nerve cells, which is what is in our brain and in our spinal cord. You have a paramecium, which is a protozoa. This is one single cell. And this thing, if you leave a glass of water with some plant in it, maybe your mother has some flowers in the house with, in the water. Maybe if you look under the microscope, you will see these things swimming in the water. This is a very common microorganism called the paramecium. Chlamydomonas, this is a unicellular algae. Saccharomyces cerevisiae, this is very important for beer and making bread. And then Helicobacter pylori, this is it's a parasite of the gut. So you can see that the shape and the behavior and the functions is very different. And you can also see the size. This is about 100 micrometer here. So this cell is actually very large, okay, because it makes many axons and dendrites. A paramecium is a bit smaller, 25 micrometer, this one here. So they are very different shapes, okay? They are very different shapes of cells and families of cells. But all living cells have a similar basic chemistry. And this, some of you may already know, this is called the central dogma in biology. We have in our cell DNA. DNA gets transcribed into RNA, and RNA gets translated into protein. So the DNA is really the genetic information. We don't do anything with it. It's just keeping information. And to get the information from DNA, you first must make a copy of DNA into RNA. This is called the transcription. And you can see here we have double strands. So we have one strand here and two strands there. So you know the double strand of DNA, the double helix of DNA. But RNA somehow is one single strand that is then read and translated into protein. So this is the very basic chemistry of life. We are going to see this into more details. Okay? And all organisms, everything that is alive, is composed of cells. Everything. Okay? Everything has cells, from bacteria to butterfly to dolphin and flowers. Everything is made of cells. And nowadays, everybody knows it. I am telling you something you already know. Everything is made of cells. But if you look maybe 100 years ago, the concept of cell did not exist. You say to somebody, do you know we are made of cells? What? They will not know. Okay? So how they started, how the concept of cell started, this is what I'm going to explain. Something that is interesting is that if you think of your cells, if you think of the cells of the trees and the cells of the bacteria, they all have evolved from the same original cell, which is the ancestor of all the cells that are currently on Earth. And about 3.8 billion years ago, this is when, more or less, the first cell is estimated to have appeared. And during that time, the evolution caused the appearance of many different cells from this same ancestral cell. We are going to see a little bit of the evolution of cells. And amazingly, the genes provide all the information for the cell shape, the function, and for the complex behavior of the cell. And this is where I go back to the origin of the concept. Very often in biology, and even now, new ideas, they come because somebody made an invention. Some engineer or some very clever person has made a new tool that is enabling you to analyze something. And the invention of the light microscope can be good by Robert Hooke in 1665. This actually prompted the discovery of the cell. Before microscope, people could not even see one cell. But since this guy Hooke invented the microscope, then people started to look at things. And there is one guy, this Robert Hooke, he created this microscope, and then he said, okay, with this microscope, I'm going to take a look at things. I'm going to take a look at my hand. I'm going to take a look at plants. And then this is cork. You know what is cork? You know this, this wine of Futane. He decided to make very thin slices of cork, and he looked at it under the microscope, and then he saw, this is very strange, these holes, and then it actually looks like that. You know what this is? Kyokai. This is a church. So you have the main church. You have Christ here, and then you have Hitoga koko ni iru ne. Here, it's the monk, Obosan no heya. Okay? These are the monks, and the name of these cells, the name of this room, is a monk cell. Like a prison cell, it's also the same. So this Robert Hooke said, hey, these things contain something that looks like cell. And this is the origin of the concept of one cell, because it looks exactly like this monk room. And with this microscope, so people started to look at many things. Everything they could put their hands on, they were looking at it. And you can see in 1880, somebody looked at one hair of a flower. This flower is called Progesantia. It's a very beautiful flower. And you can see this is a drawing that this guy has made, and where you have very nicely, you have one cell here with like black dots, but then he drew that suddenly these black dots, they sort of merge together to form something like a network of fibers, and somehow this network of fibers sort of splits in the middle and they are pulled apart, and suddenly a kind of membrane appears, and then you have something that two cells appearing. So these things were recorded very early. And nowadays, with a current light microscope, this is exactly the same thing that we can see. Okay. Do you know what this is? You have already some ideas, of course.
Cell division.
This is, of course, the cell division. And these dots that merge together, what is it?
Chromosome.
These are the chromosomes. Yes, these are the chromosomes. This is DNA that gets packaged tightly together before being equally split between each of the daughter cells. So this was great because light microscopes and the future inventions of these light microscopes, they allowed the examination of cells and some of their components. Not all of them, but the biggest component of a cell could be analyzed thanks to microscope. And people started to do these kind of things. So this is very nicely, this is the root tip of a fern. So this is the root of a plant. And scientists started to devise a method by which they could label cell structure with specific colors. For example, here you can see in the center of every cell you have the nucleus. The nucleus has been labeled red, which makes it very easy to localize where it is in every cell. This very different structure, this is what you have in your kidneys. These are the collecting ducts of your kidneys. And you can see some cells are all organized in a kind of spherical structure with a lumen, with a kind of space between them. And then you have also very different looking cells around it with blue fibers like this. So people started to see these things. And they didn't know what it was, what it's made of. It's just only, oh, this is very strange. There is a lot of organization that can be seen in the tissue, but they didn't really know what it was made of. You know, what is there? Is it, what kind of substance is it? And they also started to look at very nice things. Like you can take a look at this one. This is, of course, the division of a frog egg. From one single cell stage to, and that's 3, so you can see 2 cells, then 4 cells, 8, and so on and so forth, to millions of cells. And what is really amazing is that the very first cell is very big. And then it divides, it divides, it divides, it divides, it divides. So each cell becomes smaller and smaller and smaller and smaller, until that point where they cannot become smaller, and then the organism starts to grow. And from one single cell you have Otamajaguchi. They have just tadpoles. So this is really one single cell giving a complete organism. But if you think about this, this is really amazing. One single cell, just like a balloon, and everything is contained into this cell to make a complete organism. You are just the same in the beginning of your existence, in the beginning of my existence. We all start as one single cell. And that single cell contains everything to make you. Okay? We still don't understand exactly how it's done. There are many things we don't understand. Yeah, of course, there is DNA and the genome, but actually we don't know much. So that's a cell. Okay, if you look at with a very powerful microscope, you can see that a cell is a kind of strange shape. Sometimes that's a human skin cell. Skin cell. If you take your skin, you break your skin and you put the skin into a dish, then you see cells like that. And it has a boundary with the environment. It's called the plasma membrane. Okay, it's like the skin of the cell, really. Plasma membrane. Inside the cell, generally, is called the cytoplasm. Cyto just means cell. Okay? And then you have a very big structure, which was one of the first structure of a cell identified because it's so big. This is the nucleus. And you have different types of microscopy. This is fluorescent microscopy, and it's a pigment cell of a frog. And then you have the nucleus, same thing. And then you have the plasma membrane. And inside you have a lot of things. You have a whole network of stuff. So a cell is not just made of one nucleus and then the plasma membrane. There is a lot of things going on into the cell. So we are going to see that.
What is pigment?
Pigment, frog, they can be green, or they can be red, or they can be pink. And this color is given by a specific molecule inside the skin cell of the frog that gives them these very bright colors. So pigment is what it is. Pigment, it's a molecule that gives a color. Like, for example, in your jacket, you have blue pigment. It's not always protein. It's just a molecule that gives color. This is what means pigment. Okay? So that's the light microscope. This is currently how light microscope look like. It's a very big thing. You have your sample here, and you look at the cell with different objectives. So let's just take a look how actually it works. For a light microscope, you usually have a light source. Okay? Light comes from usually now the bottom, but it can come from the top as well. And then you need to have a special lens, which is called a condenser, which will condense light on your specimen, like a cell or a tissue. And then the light that goes through your specimen will be captured by an objective that will then that will make the light take a very specific path along its length. And then there is a magnifier lens here that will actually usually amplify it will make it bigger, really. And then you have another lens which will also make it will amplify the ray of light, and it will actually amplify the size of your specimen. And at the end, you can see whatever is in your sample, but magnified many, many, many times. Usually the eyepiece is usually 10 times. So whatever you are looking at will be magnified 10 times. This objective, it really depends. You can have 4 times, you can have 10 times, you can have 20 times, you can have 40 times. Sometimes you can have 63 times. So if you think 63 times 10 times, 630, this is a little bit as big as you can have. Over that, you are limited, because light has a limited wavelength, and you cannot magnify more than that. Okay, so this is the problem. And already, when you are using 63 times objective, you must start to use very strange technique to make sure that you catch the ray of light. So you have to use oil. So I will not go into detail with this, but essentially you are limited by light microscope because of the wavelength of light. So you cannot make things as big as you want them to be. Okay? So light microscope they were really important for people to describe generally cells and what they contain, but that was not enough. So these are different types of techniques that people can use. This is all light microscope. So you have one cell here, and then this is a kind of bright field. This is the name of a technique. This is a phase contrast. This is interference contrast. So essentially what you do, you just change how the ray of light hit your sample, the angle, where they are converged, and then you can see different things. For example, here you can sort of see inside the cell. Here you can see that the contour of the cell are well defined. You have an idea of 3 dimensions because the light is all refracted around the cell here. This one has an idea of also a little bit like 3 dimensions, but they are all kind of different. Okay? But let's say you want to see what's inside the nucleus, or you want to see what's there. You can't. This is basically as big as you can go. Fixed sample, as I told you before, so although you can look at things just with light and you have some information, but sometimes you want to make things a certain color. So you are using color to label some components. This is, for example, a section in a root. But you also have a fixative. So this is a little bit about the technique of how we observe the cell. Do you know what is a fixative? You don't know what is a fixative. Let's say you are in a hospital, or you are in the laboratory here, and you want to look at the cells of the mouse liver, for example. Because you have noticed that the mouse you are studying, they seem to be getting sick. Maybe because you have changed some of their gene, or you have used some of the drugs. And you want to observe the cells. But the problem is that if you kill the mouse, you take the liver of the mouse, and you make thin sections, because if you have the liver of the mouse, you cannot look with a microscope. You will see nothing, because the liver is very thick, right? So you must make very thin slices in the liver that then you must put on the microscope and look how they look like. But if you think of a fresh tissue, it will become bad very quickly. If you leave meat, it will become bad and it will start to rot, right? So to prevent that, you need to fix your tissue. And very often in microscopy, you must fix your tissue. And what means to fix your tissue is to do a chemical treatment so that the tissue will not be alive, it will be killed, but all the molecules inside the cells will be linked together. So you will make chemical bonds between all the components of the cells, between proteins, DNA, RNA, everything will be linked. Cells will essentially become like a big piece of plastic. It will not rot because it will become so inert that nothing will make it go bad. Okay? So this is usually used with formaldehyde. Have you come across formaldehyde? It's actually a very reactive substance that can link everything with everything. So you will link protein with DNA, you will link DNA with RNA, you will link fat. Everything will be cross-linked together. So then the tissue will be able to be kept for a long, long time. And it will become very, very hard as well, because the samples have been fixed. Okay. I will give you next time the chemical structure of formaldehyde, and you will see what it can do. And there you go, it's there. So this is formaldehyde. Simply, this is formaldehyde. It's a very small molecule, formaldehyde. And what it does, if you have a protein, you know a protein has many hydrogen. Okay, it's composed mainly of carbon, nitrogen, oxygen, hydrogen. And then if you actually mix protein with formaldehyde, First, formaldehyde will react with the protein, and then, because of this group here, you will make cross-link between proteins. All proteins will be bound to all proteins. And if you think that a cell is mostly composed of proteins, then everything will be glued together. And the protein will not degrade, the protein will not go bad, and then you can prepare your samples for the microscope. Actually, if you think of the word fixation, tsukemono, this is exactly what happens. Tsukemono, of course you are not using formaldehyde, but tsukemono is the same thing. Pickling is the same thing. You pickle your vegetables so that you are able to keep them for a long time. They will not go bad. So this process of fixing tissue with fixatives like formaldehyde, it's really the same as, you cannot keep them, you don't have fridge, and if you don't have fridge, your daikon will rot very quickly. So then you make takuan, and then you will be able to keep your takuan for a long, long time. So this is a kind of fixation as well. So very often for microscopy, you must fix your tissue, because otherwise you will not be able to look at them. You can see this very nice picture that I showed you, this frog pigment cell. Normally you cannot see that. Normally the nucleus is not blue. Normally inside the cell is not red. It's not green, if you look at the microscope. Okay? It's just without color. Right? But let's say you want to see a specific molecules within the cell. For example, in this case, the nucleus is blue. How do you think this can be achieved? How can you make a nucleus blue? Think about, maybe some of you know already, what does the nucleus contain? And how you can make it this is fluorescent, right? So if you give UV, for example, the sample will give off light. The sample will be excited and will give fluorescent light. Maybe like GFP is the same thing. Right? If you give the light at the right wavelength, you will excite GFP, and GFP will give back light that looks like in the green spectrum. But how can you do that? Do you have an idea how you can make your sample become fluorescent? How could you do that? How could you make the nucleus of a cell give off this blue light? You have a cell and you want to see, okay, does my cell have anything inside the nucleus? Do I have DNA in my nucleus? DNA is the main content of the nucleus. Then what you do is that you invent a molecule that will bind DNA. And this molecule, when it is excited by light, will emit light of a specific wavelength. And this is exactly what they did. They actually labeled DNA inside the nucleus with a specific molecule that when it is excited will give off blue light. But then, for that, you must excite your sample. And this is still a light microscope, but it is a fluorescence microscope. And you can see this is a bit more complex than the original light microscope that I showed you. This is this guy here. You have your object, okay, what you want to see, but where the normal light microscope, light was coming directly, and you could not really select what kind of light hits your sample. This is just white light. In a fluorescent microscope, you can select what wavelength of light will illuminate your sample. In this case, you can see you have green, blue, red mainly, and you have a filter that will enable you to only let, for example, blue light go through your filter. then the blue light will be reflected into your sample, and the blue light will excite your sample. And if you have anything in your sample that is excited by blue light, then your sample will emit light in return. Okay? And the light that is emitted by your sample is collected into this beam splitting mirror and will be drawn into different optics and into the eyepiece. So what you are looking at is not light going through your sample, it's light coming from your samples because it has been excited by a different type of light. So this is a very, very important invention. And what you're looking at here, it's I think these are cells in a Drosophila embryo, and you can see this green thing here, and you see this red thing here. Do you know what this is? Do you recognize anything? What it is, very difficult, but what it is DNA has been labeled by a green fluorescent label, and protein which are attached to the DNA have been labeled with a red fluorescent protein. So you can specifically label every type of molecule with a specific label. So this enables you to see maybe structures within the cell by chemical labeling or by antibody. Like, this is also kind of very important. It's a different type of microscope. It is still light microscope. You are still using photons. But let's say you have a very thick sample. Like you have a whole, 100 micrometer sample. Or you have a whole root. Or you have a big slice of whatever you want to study, like liver or something. If you look in a normal microscope, what you see in a normal fluorescent microscope, you just see nothing. Why you cannot see nothing? Why you can see nothing? If I tell you, if you have a very thick sample, if your sample is very thick, you cannot see well. Do you understand why? Do you understand why?
Because light cannot concentrate.
Exactly. Because light, you cannot get light from a specific area. So the focus is very difficult. Maybe you can focus on a specific plane, but still you will have a lot of light emitted from different layers in your sample, and you will not be able to see anything. So people have invented the confocal microscope. And the confocal microscope is very similar to that. But there is an additional thing, is that you can target this excitation light at a specific plane in your sample. So if I draw something here, imagine you have your microscope slide here, and say you have a very big cell, like something like something that is very big, with a big nucleus. With a normal fluorescent microscope, you look at everything. So if it's very thick, as you said, you will not be able to see anything concentrated. Everything will emit light. So it's impossible to see anything. But the confocal microscope, it's a laser with a very, very thin laser beam. Only one plane will be excited. In your sample, one very specific plane will be excited. So light will be emitted from one specific plane, which will be, then you will be able to pick up, and you will have a very nice focalized picture, like what you have. thanks to the fact that you can specifically target a single plane within your sample, then suddenly the structure of your sample is apparent. And what is very beautiful with that is that not only you can excite a single plane and take the picture, but then you can take another plane and take the picture, another plane and take the picture, another plane and take the picture. And then you can reconstitute with a computer the entire 3D structure of your sample. This you need a computer, of course, but this is something you can do as well. But really, if you really want to know what a cell is made of, you must use something else. What does a cell contain, beside the nucleus and the plasma membrane? You are limited again by light, because of its wavelength. So then you must use an electron microscopy. And when the electron microscopy was invented, then people started to look at inside the cell. What does a cell contain? And electron microscopy, the concept is very similar to a photonic microscopy, to a light microscopy. But instead of firing photon on your sample, you fire electrons. And here you have an electron gun that will go through a condenser lens. So the principle is very similar, and will go through your specimen. Then you can pick up all these electrons that went through your samples, and then they are gathered into the objective lens, the projector lens, and then you can have a screen or a photographic film that will then react with the electrons. So how do you think that works? This is, something completely different. That's the electron microscope. All these things are the settings and the screens and everything. And this is the kind of picture you get. This is the kind of picture you get from the electron microscope. This is inside the cell. And then you have white bits. This is kind of white background. And then you have very dense structure. What is the difference between white structure and black structure? Because of what I told you before, what is the difference? Why do you have, this is the general thing, and this is more magnified part, okay? Like, for example, what we are seeing here may correspond to this area, for example. We can see these lamellae thing here. Okay? Why this is white here? Why this is white? Why you have black dots? Why do you have big gray? Why do you have black and white? What's happening? This is an electron microscope picture, right? So what do you think happens?
Black things reflect electrons.
Actually, yeah, black things, they are dense molecular structure. So actually the electrons will not go through. Very difficult. And only when you have pieces in the cell that have almost nothing, then the electron will go through and you will have a white structure, right? So you can see around the nucleus here, and inside the nucleus, it's mostly white, but you have very dense structure. Very dense structure. Okay, so people saw that, and then they started to put names on things. So the nucleus, people knew already, because the nucleus had been observed with the photonic microscope. But the plasma membrane, now people could actually see it more precisely. Then you had this kind of very strange structure here, like this, that is magnified. Then you had this kind of roundish, a little bit looks like a nucleus, but it's a bit smaller. These were called mitochondrion. Then you have lysosome, peroxisome. So you have a lot of stuff in the cell. You really have a lot of stuff. And they all have different structure. Okay? And this is actually DNA. You can also see DNA with an electron microscope. Right. if you really magnify this structure very, very highly magnified, you have here the nucleus, and you have this thing here. And you see everything has a membrane. You have here, you have a membrane here, you have a membrane here, you have a membrane here. This is double membrane. You have membrane here. A cell is made of membrane. Please remember that. A cell is made of membranes. There are many membranes. Not only you have the plasma membrane, that is the limit of the cell, but inside the cells you have a lot of membranes. Okay? We will go on, we will see that later. Here as well, you have all these compartments, they are limited by membranes. Okay? And you always have 0.5 micrometers, so you can have an idea of the size of these things. These black dots that you have here, anybody knows? Anybody has knowledge of what it could be? I'm not telling you what is the name now of this one. Do you know? Do you know what it could be? Huh?
Ribosome.
Yes. This is actually, it's called the endoplasmic reticulum. And this is the site where RNA comes and is read by the ribosome into protein. Okay, this is the site of translation. And because ribosomes are such huge structures composed of proteins and also RNA, they are extremely dense. So then in the electron microscope, they look like really black, dense particle. So each of these particles is one macromolecule that is called a ribosome. And then people started to invent different types of things. So these kind of pictures, they are acquired by something that is called the transmission microscope, in which the electrons go through your sample. And if you have very dense structure, the electron will not go through as much, but if you have structures that are not dense, then the electrons will go through. But you have also something called scanning electron. And in this case, you will just scan the surface of your sample. The electrons will not go through, they will be reflected by the surface of your sample. And this is a little bit different. Well, I can pass the details, but this is what I told you. So the electron will hit your sample, then they will be deflected by your sample, and then you will gather only the electrons that are deflected by your sample. And what you can get is a very beautiful picture like this, where you have a very acute idea of how the surface of your sample look like. Like, for example, this is in your ear. This is in your ear. This is why you can hear me. It's because of that. So this is you have a cell here, and all these things is a very highly specialized cell that is called a hair cell, like this. And they are all tied together. So it's like my fingers, if you want. And imagine you put links between my fingers, like this, and they are all together. And this is on the surface of your ear. And it's a little bit complex. I will come to this one later. But essentially, when there is a sound, these hair will start to move. Because of the sound, these hair will move. And because of the movement, there will be an electric potential created by this movement due to specific receptors in the cell. It's a little bit complex. But essentially, this will be translated by the cell as something that you can hear. Okay? But thanks to the electron microscope, you know exactly how this look like. And this is actually what happens in just a photonic microscope. You have this hair there that you can see very nicely as a three-dimensional surface, thanks to this scanning electron microscope. The problem with that is that you cannot look at live tissue, because you must have a sample that can actually deflect electrons. So you have to use heavy metals and load your sample with heavy metals to make sure that electrons can bounce off the surface. Okay, so let's go more into the function of the cell instead of its, the tools that we can use. This is the, dimension of things. That's your finger, 2 centimeter. That's your skin and the ridges in your skin, 2 millimeter. Then if you look at the section of your skin with a microscope, then you can see your epiderm, derm, and everything. That's 0.2 millimeter, 200 micrometer. Then if you go further down and you want to see only at a few cells, then 20 micrometer. 20 micrometer, it's a little bit like the size of a cell, okay, of a normal human cell. But then if you want to look at a single mitochondria, okay, which are this very strange structure that we have seen in books before, I will tell you later what they do, then this is 2 micrometer. It's like the size of a bacteria, okay. But then if you want to take a look at what's inside the mitochondria, then you can see that they are ribosome or ribosomes. This is 0.2 micrometer. And if you want to look at a single ribosome, you remember these very dark dots in electron microscope, then you have to go down 1 factor of magnitude, so this is 20 nanometer, down to the size of molecules and atoms, 2 nanometer, down to 0.2 nanometers. So this is a little bit of how dimensions happen. And this very nice schema here actually lets you know what are the limits of the tools we are using to analyze cells. So this is the minimum you can resolve by just your eyes, okay? And then if you want to go down to the nanometer scale, then you have to use a light microscope. But beyond that, it's very difficult. So then you can go down to the electron microscope. So this is a little bit of, kind of useful things to know. Okay, so you have different types of cells. You have different types of cells. The main 2 families of cells, these are the prokaryote cells and the eukaryote cells. You are eukaryote. I am also a eukaryote. Okay? But a bacteria is a prokaryote. Die joking, E. coli, Escherichia coli, it's a prokaryote. And if you think about what it means, prokaryote and eukaryote, it's very strange word, but I think it's kind of nice. Prokaryote. What do you think it means? This actually comes from the Greek. I'm not Greek, but I kind of like to study the origin of language. So, prokaryote. Karyon means a very specific thing, and pro means a very specific thing. What is pro?
Before.
Yes. Pro means before. When you have a pro prefix in a word, it often means before something. Okay? I am proactive. When you say you are proactive, it's that usually you do things very quickly before the need arises. So you are proactive, or well, you can think of other examples. Carrion. What means carrion? Karyon means it's the Greek equivalent of a nucleus. It's the nucleus of the cell, okay, kaku. And prokaryote is simply a cell before the nucleus evolved. So essentially it's a cell without nucleus, like a bacteria, for example. But not always. Okay? You have cells without nucleus. In your body, some of your cells do not have nucleus. Which, do you know? Most of your cells have a nucleus. But some cells inside your body do not have nucleus. They used to have, when they develop, they have a nucleus, but when they get their final function, they lose their nucleus. Which means they don't even have DNA because they have lost their nucleus.
Red blood cell.
Red blood cells, yes. CKQ. They don't have nucleus. Okay? So it doesn't mean they are prokaryote. Okay? Prokaryote is just a very, very early type of cell that developed before the kind of cells that we have evolved. Okay? So you have prokaryotes and you have eukaryote. If prokaryote means before the nucleus, then eukaryote means cells that actually have a nucleus. But first, let's see the prokaryotes. And prokaryotes are really great stuff because they have very different shape, okay? And they are everywhere. They are everywhere and they are very important for the life of everything on Earth. Okay? Without bacteria, we would be unable to survive. Think of the bacteria in your gut. The bacteria in your guts are very important for digestion. Okay? The bacteria in nature are very important as well. I will let you know why. So bacteria have very different shapes, very different size, and they are the most diverse and numerous cells on Earth. And this is very simple shape of a bacteria. You see, it's completely different from the pictures I showed you before. Of course, you have the inside of the bacteria. It looks like mashed potatoes. There is nothing inside. There is no structure that you can see. It's just a soup of molecules. Okay, there is much less organization than at least at the level of the eye. Okay, there is a lot of organization, but it's molecular organization. And then you have a very thick wall around the bacteria. Our cells do not have cell wall. We have a cell membrane, but we do not have cell wall. A bacteria has a cell wall and also has a plasma membrane, a cell membrane. Okay? Plant cells have a wall, but not mammalian cells, not animal cells. Prokaryote cells are usually much smaller than eukaryote cells. But the main thing really is that there is no organelle. You know this word, organelle. It's a very strange word. An organelle it's a substructure inside the cell. Mitochondria is a type of organelle. Okay? This is a very strange word, but this is the word that is used in English to mean substructure inside the cell. So a bacteria doesn't have. A bacteria, as I told you, is just like a bag. Just looks like that. But some bacteria are a little bit more organized. Like this Anabaena, it's a bacteria that can do like a plant. It does photosynthesis. It can use light directly to get its molecules. it can build sugar just from carbon dioxide using the energy of light, just like a plant does. And then you can see some bacteria, they can associate and form kind of colonies. And they can even become specialized. Like, for example, these and these are completely different. They are the same species, but some can specialize within a community. And then some of these bacteria, you can see, have some kind of structure inside their cells with a specific function. So not all bacteria are just like Subcalis. Some are a little bit strange. But still, you have no nucleus. You have no mitochondria. You have no clear organelles that you have these kind of funny things. This is also a bacteria. What this is, you can see this is a very strange name. It sounds like a Pokemon. But what you have, this bacteria is great. Each of these little yellow dots is sulfur. This is sulfur. You know, sulfur is yellow. Sulfur is yellow. So these bacteria accumulate sulfur inside their body. For us, if you do that, you just die. But these bacteria, this is a consequence of their metabolism. Why do you think they accumulate sulfur? Why do you think they accumulate sulfur? Why? How, at the chemical level, how could they get sulfur into their body? Maybe I can tell you, I can give you a hint.
An oxidization.
Yes. Just like we get oxygen, we have oxygen in our cells, these have sulfur. Because instead of using water, instead of using H2O, these bacteria are using H2S. And they metabolize H2S, and because of that, they accumulate molecular sulfur into their body. They are completely different metabolism than us. Okay? They're in a completely different environment. But these can survive without oxygen, no problem. Without light, no problem. These are really specialized type of bacteria. And you can think of the worst environment on Earth. You know, the most aggressive environment on Earth. There will be bacteria there. At the bottom of the ocean, where you have these very hot spring going on, there are bacteria there. In the guts of cow, you know, very, very aggressive, full of methane and everything, there are bacteria there. So bacteria can colonize every type of environment where you find nothing else. But there is, within the prokaryotes, there are two different families. They are the bacteria and they are the archaea. And now I will not go into much detail on the archaea, but the archaea are considered the most ancient form of life on Earth. They are very, very old, and when bacteria, I told you, they are resistant, archaea are even more resistant. Usually when you find bacteria, you will find archaea. But where you have archaea, sometimes you don't even find bacteria. Archaea are really, really, very resistant. And the biology of archaea, compared to the biology of bacteria, it's as different when you compare the biology of eukaryote and bacteria. It's as different as that. So bacteria are as different from archaea as they are different from eukaryotes. We don't really know much about archaea. We don't really know much. At the molecular level, we don't know much. Because while they can grow in very aggressive environments, strangely in the laboratory, they cannot be cultivated because you need to reproduce this very aggressive environment in your laboratory. So this is very difficult to study archaea, so nothing much is known about them. Nowadays, people can sort of get DNA from the archaea and sequence a lot of their genes and kind of have insight on the things that they can do, but essentially, nothing much is known about these guys. And archaea, if you have, sulfur spring or hot spring or these kind of things, you will find archaea. And it's not because they are not understood that they are not important, because cows, the metabolism of cow is based on archaea, actually. Cows produce a lot of methane, because they have to digest cellulose. They are mostly eating plants, and they have to digest cellulose, but they cannot digest cellulose by themselves. In the guts of cows, there is a lot of archaea that actually digest the cellulose for the cows. So the cow really do not produce methane. Actually, this is the archaea within the gut of the cow that can produce methane. Well, this is you can look on the internet about these archaea. They are really, really fascinating form of lives, and nothing much is known. For example, in research on the biological clock, we know that there is a clock in bacteria, there is a clock in mammals, there is a clock in reptiles, there is a clock everywhere, in plants. But in archaea, nobody knows. We don't even know if they really obey the same rule as we do. There's many, many things that we don't know about archaea. Okay, so now we are going into the eukaryotic cell, and there are many, many things to see, but I think we have 5 minutes. So before starting this new section, if you have any questions, I think we can stop for here. We can stop here for today, I think. If you have any question, please don't hesitate. Yeah, maybe something I kind of forgot to mention is, although we are, this mainly focuses on our cells, mammalian metabolism and mammalian cells, but if you think of a plant and animals, they are very different, right? Plants, they can build their organic molecules from carbon dioxide. We cannot do that. We cannot make sugar. We must get it from outside. Okay? We cannot make vitamins. We have to eat vitamins. And we rely on plants to do that for us. So plants can fix carbon. They can use carbon dioxide and they can build organic molecules. But even nitrogen, plants cannot fix nitrogen. Your proteins are composed of nitrogen, almost as much as carbon. But nitrogen within organic molecules, it must come from somewhere. And it comes from plants as well, but indirectly from plants. Because chemically, a plant cannot take nitrogen from the atmosphere. It cannot. It must rely on bacteria that live inside the plant tissue that will fix nitrogen, that will put nitrogen into organic molecules, and then the plant will use these organic molecules for its own metabolism. So bacteria, they look, they are, in our mind, we kind of put bacteria on the same level as disease. Bacteria, this is not good, we have to kill them all because it makes disease. But actually, this is not true. Bacteria is probably one of the most important organisms on Earth, because we can eat thanks to bacteria. Because bacteria can really make organic molecules from anything. From anything. And it's very important even for the environment. There is nothing on Earth, almost, that cannot be metabolized by bacteria. PET bottle, they can be metabolized by bacteria. If you have oil, you have a big oil tanker who got a spill in the ocean, and you have a lot of oil in the ocean. There are bacteria who can metabolize oil. It just costs a lot of money to kind of use them. So that's why it's not so efficient yet. But there is nothing that bacteria almost cannot do. So this is really, really amazing force of organism, because of their really fascinating way to evolve. And also, but if you think, like E. coli, in the best conditions, every 20 minutes they divide. Every 20 minutes. So one generation in human is what? 50 years? One generation, 30, 50 years? 100 years? Bacteria is 20 minutes. So if you think at the speed at which they divide, it means that the speed at which they evolve is also extremely fast. So if you give a new kind of challenge, a new environmental challenge, within maybe only a few hours, a few days, some bacteria will find a way to survive with a new challenge. So bacteria are very, very important. Okay, so that's it. Maybe if there is any message of today's lecture, it's bacteria are important. Okay. So anyway, so if you have a question, feel free to ask. Otherwise, I will see you maybe next week. And if it's not interesting for you, then well, I hope you find a good lecture somewhere. Okay, that's it. You can go anytime, or you can ask a question. It's okay.